if we could hand her Exhibit 218. You recognize that item?
Yes.
And what is it?
This is my Exhibit 18A, which was a cold and cough medicine bottle. There's the barcode that is generated by the TBI and the seal that I placed on it after I was done with the evidence.
Okay. And did you perform analysis on that particular item?
I did.
And if you would, tell us what the results were of your analysis.
The outside of the bottle was swabbed. A limited DNA profile was obtained and is consistent with a mixture of at least 2 individuals, including at least 1 male. Due to the limited profile obtained and the unknown number of potential contributors, interpretation is inconclusive.
All right. So you were not able to form any kind of profile at all, is that correct?
That's correct.
So regardless of whose DNA, other profiles you would have had, there would have been no way for you to match that with anyone, is that?
Correct. It has to meet a certain strength before we do any comparisons to it, and this did not meet that quality.
Okay. All right. And can we next hand her exhibit number 200? You recognize it?
Yes, this is my exhibit number 19A, which is a Dasani water bottle. Here's the TBI barcode, and this was the seal that I placed on it when I completed it.
Okay. And did you perform analysis on that particular item?
I did.
And what were your findings?
So the mouth area and inside of the cap was swabbed for testing. The DNA profile is consistent with a mixture of at least 2 individuals. The major contributor profile is consistent with Exhibit 13A, Megan Boswell. It also had world pop stats, and the minor profile was inconclusive.
Okay. And if we could next hand her Exhibit 211. Do you recognize that?
I do.
And what is it?
This is my Exhibit 20A, which is a great value water bottle. There's the TBI barcode, my ID, and the seal that was placed on it with my initials.
Okay. And again, I'd ask you, did you perform analysis on that particular item?
I did.
And what were your findings?
The mouth and inside the cap area were swabbed in this item also. The DNA profile is consistent with Exhibit 14A, Evelyn Boswell, with world pop stats.
No other profiles on that particular item?
There was an additional allele at one of the locations, but nothing was, it was inconclusive.
Okay. Next, if we could hand her number 212. Do you recognize that item?
I do. This is my Exhibit 22A, which is a Misty water bottle. The TBI barcode with my ID and the seal that was placed on it when I was finished.
And did you perform analysis on that piece of evidence?
I did.
And what were your findings?
So once again, the mouth area and the inside of the cap was swabbed for testing, and the DNA profile matched Exhibit 13A, Megan Boswell, with world pop stats.
And next, we could hand you Exhibit 213. I'm sorry. Let me pick up what were you just handed 213?
Yes.
Do you recognize that? I'm sorry.
That's okay. Yes, it is my Exhibit 24A. It is a Food Club water bottle. Here's the TBI barcode label and my seal that I put on it when I was complete.
All right. And did you perform analysis on that particular item?
I did.
And what were your findings?
So once again, the mouth area and inside the cap was tested. Quantification results did not indicate a sufficient amount of human DNA for analysis, so no further testing was performed.
All right. And next, if we could enter 223, which should be one of the pillows. You recognize that item?
Yes, this is my Exhibit 33A, which is a white pillow. There is the TBI barcode and my seal with my ID on it.
Okay. And did you perform analysis on that?
I did.
Okay. If you would, tell us what your findings were.
So the first thing I did was screen the item with the ALS looking for saliva where somebody slept on it. And the center of the area did indicate a positive result, so that area was cut for testing. And the DNA profile is consistent with a mixture of at least 2 individuals, including at least 1 male. The major contributor profile is consistent with Exhibit 14A, Evelyn Boswell, and with World Pop stats, but the minor contributor profile was inconclusive.
Okay.
The edges were also tested on the pillow, and this DNA profile obtained is consistent with a mixture of at least 3 individuals, including at least 1 male, but due to the complexity of the mixture, it was deemed to be inconclusive.
All right. So the only profile you were able to develop was the victim, Evelyn Boswell. Is that correct?
Yes, from the center of the pillow.
We could hand her exhibit number 224 next. It also should be a pillow. Do you recognize that item?
Yes, this is a white pillow with yellow staining. Here is the TBI barcode with my ID, and down here is the seal that was placed on it after I was finished.
And did you perform testing on that particular item?
I did.
And what were your findings?
There was staining on this item, so I first tested it for the presence of blood, but it was negative. The center of the profile was tested. The quantification results did not indicate a sufficient amount of human DNA for further analysis. And the edges of the pillow were tested, and a limited DNA profile was obtained, but due to the limited profile, interpretation was inconclusive.
If we could hand her at 215 next. This be a bottle. You recognize what's been handed to you?
I do. This is my Exhibit 39A, which is a nuke baby bottle with contents. Here is the TBI barcode and the seal that I placed on it.
Okay. And did you perform analysis on that baby bottle?
I did.
And what were your findings?
So the nipple area was swabbed, and the DNA profile matched Exhibit 14A, Evelyn Boswell. And the sides of the bottle were also swabbed. The DNA profile was consistent with a mixture of at least 2 individuals. The major contributor profile was consistent with Exhibit 14A, Evelyn Boswell, and the minor contributor profile was inconclusive.
Agent Gray, I'm going to hand you an item, ask you if you can recognize that.
Yes, this is my Exhibit 9A, which is blue latex gloves with hair. This is the TBI barcode and my seal on the top.
Okay. And did you perform analysis on that item?
I did.
Okay. And what were your findings in regards to that?
Inside this bag are 3 blue latex gloves. The inside of glove 1 was a limited DNA profile of at least 2 individuals, including at least 1 male. Due to the limited profile obtained, it was inconclusive. The outside of glove 1 was a mixture of at least 2 individuals. Due to the limited profile obtained, it was also inconclusive. The inside of glove 2, a limited profile was obtained, including at least 1 male. Due to the limited profile obtained, interpretation was inconclusive. The outside of glove 2, a limited DNA profile was obtained. It was also inconclusive. Possible hairs were collected from this item but returned with the evidence because the TBI does not work hair. The inside of glove 3, a limited DNA profile was obtained. It was inconclusive. And the outside of glove 3, a limited DNA profile obtained, which was also inconclusive.
Sure. So no conclusive profiles on any of the
No.
gloves, is that correct?
That's correct.
And no, Evelyn, didn't find any DNA profiles consistent with the victim on any part of the gloves, is
No.
that correct? Okay. Judge, at this time I would move to make the gloves as Exhibit 248.
Any objection?
I guess she probably needs to, I don't think, did you open those up?
I didn't.
Okay. Do you care to open those
Oh,
up?
yeah. This is the first glove, glove 1. It has a 1 on it. And this is glove number 2. Or, sorry, that was glove number 3. This is glove number 2. As you can see, they're shredded.
Okay. And those were the gloves that you tested?
Yes.
Testified to? Okay. Judge, I think we, 248? All right, I just want to make sure. Agent Gray, did you prepare a report of your findings in regards to those items?
I did.
And I'm going to hand you a document. Ask if you recognize that.
Yes, this is a copy of my forensic biology report dated March 25th, 2020.
Is that a true and accurate copy of your report that
Yes.
you issued on that date? Judge, we move to make that exhibit number 249.
Any objection?
No, sir.
Before exhibit 249.
Now, Agent Gray, obviously you've tested a number of items in regards to this case. Is it common when you're working a case, especially a situation like this where you've got a missing child that's later recovered, do you speak with investigators who are working the case in order to determine which items are best for you to test and which might have more or less evidentiary value?
Yes, we do.
And was that done in this particular case?
Yes, I spoke with Agent Brian Fraley and Agent David Gratz on multiple occasions regarding this case.
And as a result of that, were certain items, I guess, initially you thought may not have had evidentiary value, but then later on you decided to test anyway to see perhaps they had more value?
Yeah, there were some items that weren't tested on the front end because, like the baby food containers, just because we expected if there was DNA, if it was Evelyn's, we didn't know how probative that was to the case. But later on, we did decide to go ahead and test those items.
Okay. And specifically, I want to ask you about a blanket, which I believe was your item number 36C.
Yes.
Was that in fact a blanket?
Yes.
And was that item initially not worked due to the amount of, or why was that? Now let me ask you this way: why was that item not initially worked by you?
Due to the amount of decomposition fluid on the blanket, decomposition fluid contains waste and bacteria and water, and what that does is it finds the DNA and it doesn't destroy it, but it defragments it enough that we can't generate a DNA profile. So if I were to put it on my instrument, it's so fragmented that it can't resequence it. So in this instance, it wasn't originally worked due to the amount of decomposition fluid on the blanket.
All right. Did you all ultimately make a decision later on to take a shot at it, I guess?
Yes, after speaking with Special Agent Gratz, we decided to only see if there were areas that were not stained and possibly get DNA off of that. So I tested the areas that didn't appear to have decomposition fluid on it and did not test the stained areas.
And were you able to develop any sort of profiles as a result of your testing?
It was a limited DNA profile that was a mixture of at least 2 individuals, but due to the limited profile obtained, it was inconclusive.
All right. And did you prepare a report of your findings in that regard?
I did.
I'm going to hand you a document, Agent Gray, and ask you if you can identify it.
Yes, this is an accurate copy of my biology report dated April 22nd, 2020.
All right. And is that a true and accurate copy of your report in regards to your findings on the blanket?
Yes.
Judge, I'd move to make that full exhibit number 250.
Any objection?
No, sir.
Full exhibit 250.
Agent Gray, the next item I want to talk about is the foil sheeting. Talk a little bit about the condition of the foil as you received it there in the lab.
We received it. It was heavily stained with orangish brown fluids or staining. There wasn't a lot of staining or areas that were not stained. And there were pieces that were wadded up together, but I think there were 5 pieces in total.
Okay. If we could hand Agent Gray exhibits 201 and 203. One is that white box sitting right there. Yep. You recognize those items?
Yes, this is my exhibit 15A. This was my seal down here at the bottom. And this is my exhibit, also 15A, but it was just piece 3. And then my seal is at the bottom.
Okay. And is that the foil sheeting that you tested?
Yes.
If you would, tell us what the results of your testing on those items were.
So for all of the foil sheeting, presumptive testing, chemical testing indicated the presence of blood, but presumptive immunological testing did not indicate the presence of human hemoglobin, which is a component of human blood. And for all of the stained and non-stained areas, which were 10 areas total that were tested, The quantification results did not indicate the presence of human DNA, so these items were sent to the latent print laboratory.
All right. And did you prepare a report of your findings in regard to the foil sheeting?
I did.
I'm going to hand you a document and ask you if you can identify it.
Sorry. So this is a copy of my report dated March 12th, 2021.
All right. And is that a true and accurate copy of your findings in regards to the foil sheeting you've just testified to?
It is.
Judge, I'd move to make that exhibit number 251.
Any objection? Full exhibit TBI.
Finally, Agent Gray, I'm going to, again, we mentioned the fact earlier that sometimes through communications you were asked to test additional items. Is that correct?
Yes.
And this last batch of testing that you did, there's also some additional material that was found in the trash bag, where the victim's body was recovered. Is that correct?
Yes, there was a black spoon, some Gerber baby food. One was a apple mango strawberry flavor. The other was a banana blueberry flavor. And then there was a tissue.
Kind of walk through these one at a time. You mentioned a black spoon. What were your results in regards to that item?
So the mouth area was swabbed for testing, and quantification did not indicate the presence of human DNA, so it was not taken any further.
Okay. And what about the Gerber apple mango strawberry baby food?
The sides of the pouch was swabbed. There was still food on the mouth area of the swab, so I stayed away from it just due to bacteria and focused on the sides of the swab, the sides of the pouch. And quantification was inconclusive for the presence of human DNA, so no further testing was performed.
And what about the Gerber banana blueberry baby food?
So once again, the sides of the pouch were swabbed, and quantification did not indicate the presence of human DNA.
All right. Could I see Exhibit 164? I'm sorry, 214. I'm sorry, wrong number. 214. I want to see it first, yes. Yeah, that's it. Okay. Agent Gray, I'm going to hand you what's been previously introduced as Exhibit 214. Ask you if you recognize that.
Yes, this is my Exhibit 37A, which is a tissue. Here's a barcode.
Okay. And did you perform analysis on that particular item?
I did.
Okay. And what were your findings in regards to the tissue?
So this tissue was heavily saturated in a reddish-brown stain, so I initially tested for the presence of blood, and presumptive chemical testing indicated the presence of blood, and presumptive immunological testing indicated the presence of human hemoglobin, which is a component of human blood, and the DNA profile matched Exhibit 13A, Megan Boswell, with World Pop Stats.
Okay. And using the expression world pop stats, what does that mean?
So right now the current world population is over 8 billion people, and out of 8 billion people, no one in the world has the same DNA profile as the one that generated.
Okay. So you can say with pretty certain confidence that this was Megan Boswell's profile that you found. Is that
Yes,
correct?
yes.
Agent Gray, I'm going to hand you a document and ask you if you recognize that one.
This is a copy of my forensic biology report dated October 22nd, 2021.
Okay. And is that a true and accurate copy of your findings?
It is.
That was issued. Judge, I would move to introduce that as exhibits 252.
Before exhibit 252.
Agent Gray, in addition to your responsibilities and duties within the lab there, are you also a member of the TBI's violent crime response team?
Currently, I am not. When I became a supervisor, they took me off the VCRT, so that was 2022. But in 2020, I was a team leader and a forensic biologist for VCRT.
Okay. So back in February of 2020, you were a member of that team.
Yes.
And specifically in regards to this case, were you asked as a member of the violent crime response team to perform a couple of searches?
Yes, I was.
All right. And specifically on February the 19th of 2020, did you go to Middlesboro, Kentucky to search a 2018 red Chevy Cruze?
We received the request on February 19th. We went to Middlesboro on February 20th.
Okay. And did you in fact perform a search on a 2018 red Chevy Cruze?
I did.
And what were y'all looking for?
In this instance, we were just asked to process for the presence of blood to see if maybe, since the victim had not been located, if there might have been blood in the car.
All right. And did you all find anything of evidentiary value searching through the car?
No, it was negative for the presence of blood, and we did not collect anything from the car.
Okay. And subsequent to that, did you also receive a request around February the 28th of 2020 to search a mobile home that was located at 967 Sugar Hollow Road in Blumpville, Tennessee?
Yes, we received the request on February 27th, and we went on February 28th to 967 Sugar Hollow Road.
All right. And again, what were you searching for there at that location?
At that location, we were searching for the presence of blood, and we were also searching for Evelyn.
Okay. Find anything of evidentiary value at that location?
No, everything was presumptively negative for blood, and we did not collect any evidence. VCRT did not collect evidence.
1 second, Joe. Thank you, Agent Gray. If you would answer any questions Mr. Scott has for him.
Could I see Exhibit 249, please? No, it's the report. Good afternoon, Agent Gray. How are you?
I'm good. How are you doing?
I'm good. Thank you for asking. I am currently looking at Exhibit 249, which is your report of March 25th, 2020. Do you have that in front of you?
I do.
Okay. I want to ask you specifically about the blue gloves.
Okay.
The blue gloves, there are 3 blue gloves. Is that correct?
Yes.
And 2 of those gloves had male DNA. You can confirm that.
Yes. The inside of glove 1 had male DNA. And the inside of glove 2 also had male DNA. And that was it.
And if you would explain to the jury how you determined something is male DNA versus female DNA, just briefly?
So when we see, it's called an electropherogram, which is a visual representation of an individual's DNA profile. You have your sex determining marker, which is called amylogenin. If it's an XX, that means it's a female individual. If it's an XY, then it is a male individual. And on those instances, that Y marker showed up.
Now, you talked about some of these DNA profiles not being able to be tested further. Is that correct?
Yes.
Is that because of the TBI standards for testing, or it's just impossible to do?
No, we could do it, but we've done multiple testing, and there is a quantitation limit that we know we're going to generate a DNA profile versus we're not going to get anything. So in this instance, I do believe that all of these were taken through all the way through the DNA analysis, and just it was too limited to say anything about. But mostly across the nation, we follow the same QAS guidelines, which are set forth by the FBI, so it's not just TBI specific, and they are specific into the quality of DNA before we can interpret it.
Would other labs have greater capabilities?
Not to my knowledge, is if they are accredited by the same accrediting body. They have to follow the same QAS guidelines.
Okay. So it's your testimony that no lab in the world would be able to perform more specific testing than the TBI lab?
There's probably labs out there that might do a different type of DNA that we do, but that we follow like STR DNA.
Okay. So, to your knowledge, these profiles were not tested by anyone after you got finished with them. Is that correct?
That is correct.
Okay. What about the glove, outside of glove twos, mentioned hairs were collected from that exhibit?
Yes.
Did you photograph the hairs?
No, I did not. I just documented the hairs.
Okay. Did you make a note of, are they long hairs, are they short hairs, color, anything like that?
I do believe the palm was shredded with white residue and hair on the fingers, and the hair is dark.
Okay, the hair is dark.
Yes.
But you didn't make any issue, nothing about length or anything else?
No, it was just that it was dark in color.
Okay. Let's talk about the pillow. I'm having a difficult time on this finding the pillow.
32A.
Thank you. The pillow had Evelyn Boswell's DNA on it, is that correct?
Yes, in the center of the pillow.
Okay. Could you tell if that came from saliva or what that came from?
I did not do any testing for saliva, but the area I did test tested positive for what was indicated luminescence, so it could have been sweat or saliva.
Okay. And it also had male DNA, is that correct?
Yes, it did.
Okay. And were those two mixed in the same spot, or?
Yes, it was the center area that I took a cutting from, so it would have been the same area.
Okay. Bear with me 1 second. And finally, there was a bottle, like a medicine bottle,
Yes.
found in the trash can. What number was it? Do you have that?
It was Exhibit 18A.
Yes, ma'am, 18A, that's what I'm looking for. And that item, it was tested, and were you able to determine if it had male DNA?
I was able to determine that it had male DNA, but it was inconclusive for any comparison purposes.
Okay. And as far as other DNA, were you able to determine if you could link anyone else to the bottle with DNA?
No, not, but once it's deemed inconclusive, no comparisons are performed.
If I could have just 1 minute, Judge. Ms. Gray, were you ever asked to compare any of these items to Hunter Woods or Preston Hunter Woods DNA?
I never received a standard for Hunter Wood, but I did not need it.
Thank you.
Agent Gray, I guess I'll follow up on your last answer there. When you said you didn't need it, why didn't you need it?
I didn't have any unknown male profiles to compare it to.
Okay, so there was nothing there that you would have been able to compare to anyway, correct?
Correct.
So all these male profiles Mr. Scott's asked you about were also limited. There's no way you could have drawn it. If I had submitted to you every DNA sample from every male in the world, you would not have been able to tell me
Correct.
who it was, correct?
I wouldn't even ran them since I didn't have any unknown male DNA.
All right, thank you. That's all I have.
If I could just clarify, you did have unknown male DNA on those items we just went over.
There was unknown limited male DNA, but since there wasn't strong enough for me to do any comparisons, even if they would have sent in the standard, I wouldn't have ran it.
Thank you.
That's all.
Is she free to go?
She is.
Yes, sir.
Thank you for your testimony today. You're released from this pen, then.